Hello everyone. May I start by saying how delighted I am that the UK Fungus Day is going ahead this year in what's proving to be one of the most difficult years in anyone's experience. And it's absolutely great the organisers have managed to put this together, and thank you to Nathan and the many others who've been involved in organising this, and thanks also for giving me the opportunity to speak to you. So I'm Simon Avery at the University of Nottingham, and I'm going to tell you a story about a particular aspect of our research, which has sort of interesting beginnings, which is why I chose this to speak about, and hopefully you'll learn something from this too about fungi. And we have today in UK Fungus Day, you'll be hearing a lot about many of the good things that fungi do and the things that excite people about fungi. But I'm going to tell you about the negative side, because there is a negative side also, which I'm sure you're aware of, but it's not the kind of thing that people, enjoy talking about so much, because we like to think of the nice things that these organisms do. So let's take the example on the slide of food security. And if you look to the right of the slide, these are the sort of good things that fungi do for food security, where fungi are used as food, for example, such as mushrooms and manufactured cheeses and beverages. And also many ingredients from fungi are extracted and used as food additives, for example, supplementing foods, food supplements. And all these go towards food production and enhance food security. But on the left-hand side, we have the other aspect. And there are, in fact, several ways in which fungi undermine food security. So first of all, we had some very serious crop pathogens, which devastate crops across the world and undermine our food supply chains. Food spoilage fungi also. So once food has got to supermarket shelves, into our homes, of course you'll have noticed if you leave things lying around too long that you'll get spoilage. And so this contributes in a big way to food waste. But they can also be dangerous. So several fungi, which you can find in foods, produce toxins. And so this is unsafe for human consumption. And some fungi, of course, are also infectious, so can cause disease, and those might occasionally be associated with food also. And so these all contribute to food insecurity. So there's both this negative and this positive side. We can look more broadly at the kind of problems that fungi cause and some of the figures associated with this. So it's been estimated that fungal infections kill over 1.6 million people per year, which is a huge number when compared with other major disease forms that we're kind of quite familiar with, and probably more familiar with from the press. But this is the kind of impact that fungi have. I mentioned plant pathogens already. These are the organisms that can destroy crops, so potentially up to 20% of crop products, depends where in the world and the types of infection. But in total, enough food annually to feed 600 million people, which in an age when we are increasingly concerned about preserving food security for the future, this is obviously a major challenge that needs to be addressed. And this is why the global fungicide market, fungicides deal more with controlling fungi that infect crops or grow on objects around the house or outside. So this is one way in which they're treated with fungicides, and this is an enormous industry, so more than $7 billion annually. Antifungals, this usually refers to drugs that are used to combat human disease, and food preservative these also add to these fungal control industries concerned with generating chemicals that enable us to control fungi. So how do we control fungi? And indeed it is mostly with these sorts of chemicals. So antifungal drugs for human disease, fungicides, as I've mentioned, and also food preservatives. On the right is just a picture from my laboratory which shows some growth of fungi with different dilutions of organism forming colonies on agar. So there are different dilutions. You see individual colonies over here to the right. And you can see that when we add these different agents, the identities aren't important really. In this case, we can do things like combine agents where we put two together and it has a much more significant impact on fungal growth at the bottom here than either one alone. These are ways to help enhance the activities of some of these agents. You combine them together, they have a stronger effect. But still, there are a number of disadvantages with the use of these chemicals. So, of course, there is resistance. We hear a lot in the press about antibiotic resistance. Well, fungi also do this, antifungal drug resistance, fungicide resistance, and the more you use these things, the more the resistance spreads. But of course, there's also concerns about the environmental impacts of using active chemicals. These are agents which can have side effects in other organisms in the environment, be transferred through food chains, and so there are environmental impacts concerned with that, and of course, therefore, regulatory restrictions. And these regulatory restrictions tend to tighten over time, and so the concern grows as time moves on. So, are there ways to find alternatives to control fungi, that is, without using chemical actives? There's a lot of interest in the moment in the use of natural products. So these are natural extracts from plants, for example, or other organisms. But this can also involve, of course, active chemicals. Many of these natural products are also active chemicals, so it doesn't really fully alleviate the concern. In crops, there are crop cultivars which are naturally or have been bred to have genetic resistance to infection, and that's really good. So that avoids using fungicides, for example, or reducing the use of fungicides at least. But fungi then can be in combat with those crop cultivars and develop resistance to that resistance, and so there's this kind of evolutionary warfare going on. So these aren't foolproof measures. Biological control is another area depicted by some of the images at the bottom of this slide, where you grow organisms together and one can inhibit the growth of the other. And this is in fact similar to the way in which antibiotics were discovered in the first place. And there are methods to try and control fungi. They're difficult to control, to implement, but there are some developing procedures here which hold some promise. Another way potentially is blocking fungal attachment. Attachment of fungi to surfaces is the prelude to really most of the problems that they cause. A fungus needs to attach to a plant surface in order typically to cause most types of plant infection. In humans, they need to stick to surfaces on our body, inside our body, to be able to spread, penetrate tissues, organs, spread through the body. And also materials deterioration around the home, for example, they need to attach to surfaces in the first place. So if there was a way to control fungal attachment, that first attachment phase of infection or colonization, could this be an alternative way to control fungi? And this is the story I'm going to tell you briefly about over the next few slides. So how does this kind of project develop? I didn't have a prior thought that we were going to focus on fungal attachment. It arose through a similar way as many research angles develop, just sort of by chance really. So this is a bit of an aside from the science. One of my pastimes is cycling, and I've got a few friends I go cycling with, as shown here. I'm second from the right on this picture. That's having ascended one of the alpine passes. And far right is a friend, Ricky Wildman, who also happens to be a chair in something I don't understand at fully at the University of Nottingham. So he's a chair in multi-phase flow and mechanics, and it would seem there's very little commonality in our interests. And indeed, we sort of thought that was the case too, until one of our cycle outings, we just started talking a little bit about what we do, just, you know, to pass the time, and we sort of struck upon a potentially common area of interest. And this revolved around a colleague of Ricky's at the time, also at the University of Nottingham, Morgan Alexander, a chair in biomedical surfaces. And he put us in touch with each other following our conversation while on our bicycles. And one of the interesting things about Morgan Alexander's work and the work he'd been doing with Ricky is their development of a platform which is quite unique to Nottingham, involving materials screening. So finding new materials which do new things and can be used in different ways, including in biological applications. So I went to see Morgan, and he told me about his platform. And this is a little bit of what it's about. I won't go into too much detail, largely because I don't understand a huge amount of the detail. But this is their high throughput screening platform for polymer materials. So polymers, you think of, you know, plastics and things like this, everyday items that are polymers. There's many other examples too. They're made from monomers, which are the building blocks basically, and there's various processes you can undertake to turn these into polymers, which are basically sticking these chemical building blocks together. So in this case, ultraviolet irradiation, for example. And in Morgan Alexander's lab, they can do this, but they can do this in very high throughput. So they can make lots of different material polymers. So they can produce what we call microarrays of polymers, in this case methacrylate polymers, if anyone's interested in chemistry. This is a picture of one of these slides, just to give you an idea. It's about the size of a microscope slide, if you're familiar with microscope slides, so a few centimeters in length, basically. And they produce these microarrays, which are hundreds of different polymer materials. Their ability to do this and produce them in this microarray format produces this unique screening platform where you can then screen these materials for properties of interest. And of course, our interest was, and which I discussed with Ricky and Morgan as my developing collaborators, was in this fungal attachment problem. Because fungi, of course, attach to surfaces, or we could call them materials. Could we find materials to which fungi couldn't attach? And could we use this screening platform of Nottingham's to try and find such materials and then exploit them? So we embarked on this research endeavour, and one of the major advantages of this type of approach is it's to try and find materials which passively block the fungal attachment. So they're not actively killing the organisms. And from a sort of legislation, environmental concern, etc., that's far more appealing and far easier to use in a way that's acceptable to people and industry. If we can find something that just physically, passively blocks the binding of organisms to surfaces. So we looked at two organisms. Over here on the right, C. albicans, that's Candida albicans. I'd imagine most in the audience would be familiar with this organism, will have heard of it. Candida, of course, is a major human pathogen, well known. In fact, we all are quite prone to carrying Candida. About 50% of the population will have Candida, perhaps me too, as a natural commensal. Commensal means it's not really doing much, it's sitting there as part of your natural microflora. But of course, when things go wrong, people are immunocompromised, etc., they can develop very serious and potentially lethal infections. And so we're quite familiar with Candida. The other organism we screened was Botrytis cinerea, which is a serious crop pathogen. So these are the two organisms we screened against these polymers. And I won't go into detail in these sorts of images, but this is a measure of fungal attachment up here. So 100 is kind of the average, just what we expect most materials to do in this case. But among the many hundreds of materials that we screened through the platform, we found some to which Candida or Botrytis could stick more readily. So these are the ones that score over 100, but also some to which they attached quite weakly. And down here, right down the bottom, we have materials to which, a minority of materials, but the odd material to which Candida or Botrytis really cannot bind at all. And this is a sort of depiction of what it looks like under the microscope. So here we have 3 example polymers. To the left is a material to which the fungal spores are sticking quite happily. You can see lots of fungal spores. Next one along, a material to which they don't bind quite so strongly, but there's still adequate binding, whereas here there's almost no attachment. So in this case, this is Botrytis cinerea spores. We get the odd one, but almost no attachment to this particular polymer material which we found in our screen. So this is one that is of interest and we want to take forwards. And we looked at other fungi too. So I've mentioned Candida albicans, Botrytis, another plant pathogen, Zymoseptoria trititii. This is associated with serious infections of wheat, which is obviously a major crop across the world. So means to control Zymoseptoria trititii are very attractive. And we also looked at Aspergillus brasiliensis, which is what's regarded as an environmental fungus. It's the kind of fungus that if you sample spores from the air and identified them, there's a good chance Aspergillus brasiliensis may be among these. And how much overlap was there in the results? So it's quite interesting because Candida we supplied as cells in the screen. This is vegetatively growing cells of Candida, whereas Botrytis, there was more the dormant form that we could use for the screening platform. So there were spores of Botrytis. And Candida and Botrytis, in evolutionary terms, they're both fungi, but they're within the fungi, they're quite removed in evolutionary terms, as in they separated a long time ago through evolution, and they're quite distinct organisms. But despite that, among the materials we looked at and found to be of interest, 27 showed low attachment by both of those fungi. Some of them were specific to Candida, some to Botrytis, but some to both of those fungi. So that's of some interest because it means some of these materials can resist not just one fungus, but more than one fungus, and potentially others too. We were interested in understanding how these findings could be used in manufacturing ventures. And I mentioned that my colleague Ricky Wildman was a chair of the multi-phase flow fluid dynamics, but he does a lot of additive manufacturing, which to me is a sort of 3D printing. And so we found that actually certain of our HIP polymers could be 3D printed. So here are some examples. This is you'll recognise this image, Twitter bird. This is University of Nottingham logo. Here are just some coupons, which are like, could be like pills, which could be medicines, for example. And over here on the right, this is a voice prosthesis part. This is the valve, in fact. And voice prostheses are interesting because Candida is a major contaminant of voice prostheses, and patients who have voice prosthesis implants, it's about every 6 months they need to be replaced because of Candida biofilms forming around this valve piece. So if you could have a valve which resisted the attachment of the Candida, then it would be possible, potentially, to avoid this very invasive, costly replacement of voice prostheses every 6 months, which, as you can imagine, for people who have voice prostheses, this is a really unpleasant experience, and it's very costly too. So we discovered that one of our polymers we could print. It was suitable for printing purposes, and we could reproduce this valve structure using our polymer. So of course the question then becomes, these things that we're printing and making, can we use these potentially to resist attachment of organisms like Candida in practical applications? So here, with regard to Candida, of course, we tested this. Here are two valves from the voice prostheses, and the purple here, we've got a purple dye which picks up the Candida, and as you can see, the control valve, which we got from manufacturers, we have a good industry link on this project, and we see a very avid biofilm formation by the Candida. But here, when we manufacture by polymer, we get a much lesser growth or attachment of biofilm. And here, we just need to take this this valve flap and just sort of mix it a bit with a bit of water, and that biofilm will fall off. It's loosely associated with the valve flap, unlike the control valve flap. So biofilm formation by Candida on our manufactured valve flap is strongly inhibited, and we've done lots of replicas of this shown on the left. So there's potential application there. We also looked at plant infections, and this is with Botrytis cinerea, and these are lettuce leaf discs. So we go down to Sainsbury's, buy some lettuce, punch out some discs. These are sort of 2 or 3 centimeters in diameter, and we introduce spores into the center of the leaf discs. We do this on day 0, so on the far left you of course can't see the fungus yet because it's just in spore form that we've added it and it hasn't started growing out yet. But here on the top we have the control, and this is exactly what we expect to see. Over the course of 3 days we start to see the fungus growing until eventually we have this lesion, quite marked lesion, by day 3. On the bottom, we have pre-sprayed the lettuce leaf discs with one of our HIT polymers. So a quick spray and then add the Botrytis, and we see that after 3 days there's no sign of infection. And again, we've repeated this many times with different polymers, and these are the sorts of results we get. And so this opens up the possibility of potentially spraying crops with these types of materials to help them resist infection from the types of fungi which can initiate infections on plant surfaces. So what's happening with this work now? And this is just to show you the sort of timeline of how these things can develop and where we go with them in the future, in particular where there's some potentially commercial interest in applications of this sort of technology. So from the initial idea, which I think that bike ride photo and conversation around then was about 2016. It takes a bit of time obviously to do these sorts of experiments, especially when there's lots of collaborators involved, cross-disciplinary from different universities. But we initiated engagement with industry about a year ago or so. So we started having conversations under confidentiality agreement. We got quite a strong response, so industry is interested. Diverse arms of industry too, so agrochemical industry as well as medical implant type industry, catheters, voice prostheses, etc. We filed, so because of the interest and the potential application, we filed a patent application earlier this year. And this had to be, of course, before the paper was published, otherwise we'd have shot ourselves in the feet. So the paper was published subsequently, we made sure the patent was done first. And a press release coincided with that paper publication. This is the sort of headline of the publication that came out in June. You could look it up, it's open access if you want to look up science advances. As you can see, the people I initiated the conversations with are here, so Morgan Alexander, Ricky Wildman, as well as a whole host of other researchers who were introduced to the project, because this is a really multidisciplinary project, and it's one of the attractions for me, because a lot of research is going this way these days, and it's where you can really make some quite significant advances. On that author list, there's only 3 sort of fungal biologists: Cindy Valliers, Catherine Davies, and myself. The remainder are from diverse arms of science or analysis, and we all came together to produce this. So the press release, and following that, there was things like interviews on the radio, and one of those was turned into a podcast, which I've shared, and I think should be available coming straight after the end of this presentation, which is in a few moments' time. So this is where I was quizzed on the sort of validity and potential applications of this type of technology. And so, I'd encourage you to listen to the podcast. I think it's 5 or 6 minutes long, and it might be asking some of the questions you might be thinking of yourselves. And just earlier this month, we've heard that we've had fairly significant grant funding awarded from the BBSRC, which is one of the major UK funding bodies from the government. And this is a particular scheme which is follow-on funding. So it's where a piece of sort of fundamental research has potential application. So it's for this funding is for translation of that research into practical application, including commercialization. That's what we aim to do through the 2-year funding period that will probably start in January at this stage. So some conclusions from this. Obviously, some fungi have lots of great things about fungi. This is why we love fungi, but also they've got this dark side. So they're major pathogens of humans, crops, spoilers of foods, or household and other materials. So you see them growing around your house, on your food, and these have major impacts on health. Food security I focus on a little bit, and industry, the sort of economics underlying industry and success of companies can hinge on these sorts of things also. Strategies for fungal control are needed, and those that avoid chemical actives, this is the traditional way of controlling fungi as chemical actives, and I gave examples of alternative methods. There are other ones potentially in development, but there are other possibilities besides those I mentioned, but these are highly sought at the moment to find ways controlling fungi without chemical activities, which pose these problems I highlighted. And materials that passively resist fungal colonization could be one solution. And I didn't mention it, but part of our work we did show that indeed this was a passive effect. They are not killing the fungi, these materials. And a final line that, as I said at the beginning, for many things that we do can crop up many ideas when you sort of least expect them. You could be talking with people who might have no interest in this sort of area at all. Apparently fungi, for example, biologists, but you can go totally outside those fields and find commonality. And this is the way a lot of research is going these days, where people join forces on their different fields of expertise and find a way of harnessing that in a new way that hasn't been done before to find something new. And I think this is a really exciting thing. So to follow this, there is a podcast if things work as I expect, and the podcast will pose some of the questions, as I said, I expect you might be asking, such as safety of the use of this sort of technology. And one thing I will add, I'm not sure I say it in the podcast, but there's concern about plastics, and people think of polymers and plastics, and sort of spraying this in the open field might raise some of those concerns. But thanks to colleagues in chemical engineering, etc., there are ways of manipulating these sorts of things. So the biodegradability versus the resilience of these sorts of materials can be adjusted through chemical modification. And so there are answers, and there are precedents for using these sorts of materials for other purposes, not passive control of fungi, but for other purposes, both in the field, so on crops, for example, and also in humans. There's a long history of using these types of materials for other purposes, including clinical trials, etc., in humans. So safety concerns have largely been addressed, but of course these are issues we're very conscious of as we try to progress this research into commercial application. Thank you very much for listening. I hope it was of some interest, and really just to highlight the other side of fungi, besides the side we love, but also this poses serious research questions and challenges which people are actively trying to address, and it creates for some quite good stories. Thank you, and I hope you enjoy the podcast too.